So thank you so much for the invitation to speak today. And I'm really excited because we actually just found out today that this work has officially been published. So if you go to my Twitter, I have it pinned on the top if you're interested in looking more into it. And so just a brief overview, I'm gonna, encourage you guys to study sex differences in everything that you're doing. And then give you a little snippet of what we've been doing in this project with 4 core genotypes data in the hippocampus. So I'm really excited that Dr. Gunn kind of introduced it to you guys. And so why do we think that it's important to study sex differences? And the first, which is probably why everyone started doing this, is because it is a requirement. So if you want to get funded by the NIH starting in 2016, they said, yes, you need to include both males and females, which is great. And you can see here just by the number of publications that cite sex differences, once they required it, you had almost a doubling in the number of publications that are including sex differences. But still, when we're going to look at publicly available data, less than half of that data still even annotates for sex. So make sure you put that in there. Also, we want to make sure that there's sex equity in healthcare in general. And so there's this really good book by an ER doctor, which talks about how, physicians are being trained based off of male data. And so even a woman that comes into the ER and is having a heart attack, it presents differently. And so women are not being given equal treatment as men are in medicine. And a lot of this has to do also with, hormones and female biochemistry, and also the fact that females are more likely to be prescribed hormones such as birth control. In addition, and more from a basic biological perspective, we can actually harness these natural genetic diversity between males and females to understand why there is sex differences in aging processes and why biological sex may be conferring risk or resilience to different age-related diseases. And outside of, chromosome aneuploidies, biological sex is the greatest genetic difference between any 2 individuals of the same species. And yet it is commonplace for the sex chromosomes to be completely removed from GWAS studies, even ones that are directly related to biological sex. And so when we're looking at male-only data, male and female humans actually only have about 98.5% genetic similarity. Whereas if you look at the similarity between humans and chimps of the same sex, they have 98.8% similarity. So I would prefer data from female chimps as opposed to male humans. And if we're talking about sex hormones, it's completely different between male and female humans. So again, just really important that we're studying both sexes and considering these large natural biological differences between males and females. And when we look in the brain, this is a study previously out of Dr. Freeman's lab, my predoctoral mentor. And this is whole tissue transcriptomics from the hippocampus of mice with aging. And what we see is a sexually divergent phenotype where we're only getting differences in old males and females. And most of these pathways are involved in neuroinflammation, microglial mediated pathways. So we think that there's this intersection between both aging and biological sex that is really important to understand in the hippocampus. And this is something that several speakers have talked about during this session. And so as we're thinking about this basic biology, what is actually causing these sex differences? Well, we can have hormonal differences from the gonads, either testes or ovaries. But then we also have these genetic differences, whether there's Y chromosome, X inactivation. And so although sex is determined genetically, when people are thinking about sex differences, they're generally focused on the hormonal contributions. As I said, sex determination is genetic. And so there's a single SRY gene that will trigger the testes determining pathway. And then sex differentiation occurs as a result of gonadal hormones. And so this was kind of spoiled with Dr. Gonstock, but how can we uncouple these so we can understand what are the separate contributions of gonadal hormones and chromosomes to sex differences in phenotypes across the lifespan? And so we can use the 4 core genotypes mouse model, where the SRY, testes determining gene that's normally on the Y chromosome, is removed from the Y chromosome and inserted onto an autosome. And so in that way, we can uncouple genetic and chromosomal sex, and we'll have XX mice that have either ovaries or testes, and XY mice that have either ovaries or testes, as Dr. Gon described. And this gives a really elegant two-way design where we can study the separate contributions of sex chromosomes and gonadal sex to these sex differences in phenotypes. In a really nice paper out of Dina Dubal's lab, they crossed the 4 core genotype mouse model with a transgenic model of AD. It was a HAPP model. And what they found was that in the AD mice, XX mice survived at higher rates than the XY mice, with no difference by gonadal sex. And that also the XX mice tended to have better performance on cognitive behavioral tasks than the XY mice. And so the sex chromosomes in some way are contributing to the resiliency or sex differences in this AD mouse model. And so that led me to the question, what are the contributions of gonadal and chromosomal sex to the sex effects that we have observed in the mouse hippocampal transcriptome and also the epigenome? And so what we did was we took adult 4 core genotype mice, dissected the hippocampus, and from whole hippocampus isolated DNA and RNA for epigenomics, so whole genome oxidative bisulfite sequencing to look at DNA methylation, and also RNA sequencing to look at the transcriptome. And so I don't have time to dive into the entire paper, but as I said, just came out today. And what we found, we first looked at the sex chromosomes. And so most people, when they're thinking about X inactivation, they think X inactivation just balances gene expression between males and females. And it's not actually that simple. So there was a subset of genes that are more highly expressed in the XX genotypes on the X chromosome, including things you expect to see like exist, and genes that consistently escape X inactivation. But then there were also a subset of genes such as TLR7, which Dr. Gon briefly mentioned, that were actually more highly expressed in the males or the XY genotypes, regardless of their gonads. There were only 2 genes that showed a gonadal effect where they had higher expression in the XY genotype or the males with testes. And interestingly, one of those was ACE2, which is the receptor that COVID-19 uses to enter the cell. And then of course, the Y chromosome genes, there's very few that are expressed in the hippocampus, but those were only expressed by the XY genotypes as expected. And so the sex chromosome differentially expressed genes were regulated mostly by chromosomal sex, which was not all that surprising. And they were enriched in histone modifier pathways, specifically with H3K4 and H3K27 demethylases. And when we looked at the DNA methylation patterns across those developmentally programmed histone modifications on the X chromosome, we saw large differences between the XX and XY genotypes, indicative of X inactivation. And generally, when we think about the relationship between DNA methylation and gene expression, we think of an inverse relationship where high levels of methylation within a promoter region are repressing gene transcription, and hypomethylation allows for transcription factors to bind and that gene to be expressed. And this is exactly what we saw on the X chromosome, where we have this strong negative correlation between gene expression and DNA methylation. And so how may the sex chromosomes be contributing to aging phenotypes? Well, as suggested by a question right before I started, there may be some variable escape from X inactivation. And in my paper, I see that there may also be an interaction between gonadal hormones and the X chromosome inactivation machinery. There may also be some spreading of the X inactivation machinery onto autosomes, and then also mosaic loss of the Y chromosome. When we look at the autosomal transcriptomics, what we see is that most of the genes that are differentially expressed, both by chromosomal and gonadal sex, are involved in important inflammatory pathways like antigen presentation and interferon response. And their separation both by sex and by chromosome. And so when we tried to relate this to our DNA methylation, we didn't see that strong negative relationship that we expected to see. And that was likely because the cell type that is primarily responsible for producing these genes is a minority cell type. And so our next goal is to sort microglia and look at the epigenomics and transcriptomics relationship in the specific cell type. And so some quick conclusions. Please annotate sex when you're depositing your public data, and don't throw out the sex chromosomes because they could be involved in whatever sex differences you're identifying in your studies. And so with that, I'd like to thank my pre-doctoral mentor, Dr. Freeman, and also my new postdoctoral mentor, Dr. Mike Stout, Dr. Steve Austad, and Dr. Jessica Hoffman at UAB were the reason that I was able to get these wonderful 4-core genotype mice. And so we have highlighted here also other co-authors on the paper, and of course, funding sources. So I'll take any questions.
Hello.
Hi.
Hi. Super cool work. Thanks for that. I just wanted to know, how do the 4-core genotype mice do, like anecdotally? Like, do they have any other phenotypic differences besides sex presentation, like behavioral ones?
So I haven't personally done any behavioral testing on them. The only thing that I know is that the XY females have a stronger inflammatory profile.
Did you do hormone measurements like estrogen and testosterone on these? And then also, are they sexually dimorphic, even with their differences in, I guess, the 4-core? Like, do you still see the testicle presenting, I guess, males looking larger than like their corresponding females and within their group?
So I did not take hormone measurements on this cohort of mice. Dr. Freeman and Dr. Stout actually flew to UAB and collected tissue and then brought it back for me. So I didn't have those mice. I can tell you that the X males have hypogonadism. So they have very small testes compared, and we're trying to figure out how we can resolve that so that you don't have to do gonadectomies and you still get, you know, similar hormonal profiles between them.
Did we have time for another one?
I think we should move on.
Okay.
Thank you.